This conference will now be recorded.
Yeah, immunology has a huge overlap with other subjects. It has an overlap with pathology. It has an overlap with pharmacology. It has an overlap with rheumatology. It has an overlap with microbiology. So basic structure of immune system and the process by which it gets activated and the process by which it kills the organism that is entering is what is dealt in physiology. And further details of this is taken in second year of yours. Microbiology, there is a complete unit on immunology. Yes. So it is taken for around 15 to 20 hours, immunology. So here today we are going to discuss in 1 to 1.5 hours just about the basic, what are the basic processes taking place inside the human body. First, to protect the body from entering pathogens. If the pathogen enters, what are the processes taking place inside the body to protect the body from that pathogen? And you can very well relate to this with all the recent this thing that you have gone through, and you will very well relate to this with your day-to-day life. So let us now start. Let us now start with an analogy. I'll request you all to take a paper like this, so that we'll make a flow chart over here, and we do not disturb that flow chart in the between. Whatever information regarding the terms being described in the flowchart is given, please write that description in some other book or paper side by side. So don't write the information, the description regarding those things in this paper. In this, only write the flowchart. The other information that I am giving, the dictation part and all the those things, you can write down in your other book. Okay. So let us now start. Let us start with immunity as such. Just a second. Let me adjust this. Yeah, let us take an example. Let us take an example over here to understand. For example, our country. This is our country. This is Jammu and Kashmir. Okay. This is your Punjab. How am I drawing is? Yes, this is your Rajasthan. This is your Gujarat. Okay. Now this is your South India. Okay. This is your Northeast. Okay. Here, Northeast. Here is our favorite country. This Pakistan. Yes. Now, from Pakistan, some soldiers of Pakistan, or let's say terrorists, I'm not against any country, nothing like that, but just to understand, we are dealing this. Yes, we are some soldiers of Pakistan, some terrorists are wanting, they are wanting to enter inside our country. So what are the processes that first we ensure so that they should not enter inside? So first thing that we have are your borders, your barriers. So first thing you write, barriers. Okay, the first thing is barriers. If they are able to cross the barriers, there should be something inside the barrier that should be stopping them. So that is your local soldiers. These are your local soldiers, guys. These are your local soldiers. I hope this is visible. These are your local soldiers. We are going to have an analogy of how our immune system is related to that. These are your local soldiers. Okay, yes. Now, these two are the components of your innate immunity, guys. Innate immunity. Yes, innate immunity. Now, if barriers and soldiers, if the bacteria, if the terrorist is coming inside, the barriers are not able to stop them, the local soldiers will kill them locally. Okay, if the local soldiers are not able to kill them, then the innate immunity has failed. The local defense has failed. Now this has to be dealt in your central government, dealt by the central government. So the information has to be given to the central government, guys. Yes. Now the central government is the Ministry of Defense. Yeah, Ministry of Defense will get activated. After the Ministry of Defense get activated, after it analyzes and recognizes that certain threat is inside the country, the terrorists have entered inside the country, the Ministry of Defense will activate whom, guys? Whom they will activate? They will activate either the army, guys, yes, or the navy. either the army or the navy. The first thing they'll do is they'll recruit the army from all parts of the country towards the place where there is infiltration by the organism, infiltration by the terrorists. Yes, so this is recruitment. Now, when will the Ministry of Defense call the army, and when will it call navy? Depending upon the organism, depending upon the terrorists. If they are coming from the navy side, if they are coming from the seaways, then they will call navy, and if they are coming from the roadways, they will call army, and if the attack is by airways, they will call your air force. Yes, this is all that is happening. So innate immunity has now failed. The bacteria, let us talk in terms of our immunology now. The bacteria which was trying to enter inside the body was first prevented by your barriers. Yes, we have written the barriers. Now, what are the barriers inside the body? What is there? We are going to discuss it detailedly. So first, let us see that there are anatomical barriers, some anatomical barriers and some physiological barriers. So these are the two barriers that we have. Yes. Now, then the local soldiers, guys. We can compare these local soldiers with the local inflammation that is taking place. Once the organism enters inside, there will be local inflammation, guys. Local inflammation. So this local inflammation, what it will do, guys? Is it visible? Is it blur? Yeah. Yeah.
It's quite
What?
blur.
I'll write
Yeah,
a little big. Yes.
yeah, yeah.
What this local inflammation will do? What is the purpose of this local inflammation, guys? This local inflammation will bring cells of innate immunity, proteins of innate immunity towards the site of infiltration, guys. Yes, write down somewhere else what these cells and proteins are going to do. They are going to bring cells and proteins of innate immunity towards the site of infiltration, so that even if the barrier has failed, they can locally destroy the bacteria, and do not allow it to systemically progress in the disease, guys. Same applies to your coronavirus, guys. The coronavirus entered your respiratory epithelium. Now, there will be barrier. What are the barriers in each of the tracks that we are going to see now? Yes, what are the anatomical and physiological barriers? Then what happens? There will be local immunity, guys. Local immunity in the form of your cells and proteins. What are those cells and proteins? We are going to see in very detail. If that does not take place, if these two fails, that means your innate immunity has failed. If innate immunity has failed, which immunity does this go activate now? If innate immunity is failed, we have to now activate your which immunity? Let me know in the chat box. What are the two immunities that we have? Major groups of immunities: innate immunity and let me see in the chat box. Yes. Yeah. What are the answers I am getting?
Yeah, acquired.
Acquired immunity, guys. Acquired immunity. So innate immunity is your local immunity. Acquired immunity is your systematic immunity, guys. So the activities that are taking place once the innate immunity fails to activate the acquired immunity, this is known as activation, guys. This is known as activation of innate immunity. Activation of your acquired immunity. Acquired immunity, guys, is also known as adaptive immunity. So this is full-fledged, well-organized, well-coordinated system which works to protect the organism. Now, this will also have memory, guys. What are the differences between innate and adaptive immunity? That we are going to discuss in a while. So first, let us discuss what are the changes taking place, how the adaptive immunity is first getting activated. Infiltration occurred, barriers failed, your local immunity failed. So one of the soldiers from the local soldiers, one of the soldier, what he will do, he will take the photograph of this infiltrating terrorist, photograph and some ID cards he will take. He will take that ID card and show it in the central government Ministry of Defense, saying that someone has entered. This is his identification. Now, this person who is presenting the information regarding the pathogen to the central government is known as your antigen presenting cell, guys. So this soldier is your antigen presenting cell. Now, what is the meaning of antigen presenting cell? What is the definition? I will tell you when we are going to discuss that. Let me first give you an overall review what we are going to discuss. Now, so the innate immunity, once it failed, the antigen presenting cell will now alert the acquired immunity. It will activate the acquired immunity. Now, this Ministry of Defense which is activated, Ministry of Defense in the central Delhi. Okay, Delhi. Now, what can this be compared to? This soldier we have compared to APC. These barriers we have compared to this thing. These local soldiers we have compared to cells and proteins. Now, what can the Delhi Ministry of Defense be compared to? It can be compared to T0 cells, guys. T0 cells. Okay. T0 cells. One more thing that I would like to say here only is you have, what are the blood cells that you have? NEB and LM, NEB and LM, your granulocytes and agranulocytes, neutrophils, eosinophils, basophils, lymphocytes and monocytes. Now, what are the cells responsible for innate immunity, and what are the cells responsible for acquired immunity, guys? Yes, what are the cells required for acquired immunity? Let me know. Type in the chat box what are the cells required in acquired immunity, your adaptive immunity. Rest all will be there in the innate immunity. So may I have the answer? Am I getting any answer?
Yeah,
cells.
T cells,
Yeah.
they say.
Not only T cells, guys, your lymphocytes,
Yeah,
lymphocyte consists of your B lymphocytes and T lymphocytes, guys. So this, along with your macrophages, is in your adaptive immunity, guys. Adaptive immunity. And neutrophils, eosinophils and basophils along with your macrophages is your innate immunity cells. So one information you got, the cells that we were talking about here, what are these cells? These are these cells: neutrophils, eosinophils, basophils and sometimes macrophages. Macrophages is a major part of your adaptive immunity, but the sole cells of your adaptive immunity are your B lymphocytes and T lymphocytes. Let me now once more ask you a baby wala question. What are B lymphocytes participating in? What is T lymphocytes participating in? In acquired immunity, we have two types of immunity, the humoral and the cell-mediated immunity. Now, B cell is participating in what and T cell is participating in what? Let me know in the chat box. Yes. Let us be interactive. Let us not make this boring. Yes.
Yeah, everyone says B cell in humoral immunity and T cell in cell mediated.
T cell in cell mediated immunity. This is a wrong concept. This is a wrong concept. T cell is involved both in humoral as well as cell mediated immunity. Now what this guy is telling, we all learned that B cell is responsible for humoral immunity. Yes, B cells are the main cells of humoral immunity, but to activate the humoral immunity also you need T cells, and T cells are also a part of your cell mediated immunity. Let me tell this more clearly. More clearly, let me tell. So these are your T cells. Okay, I will write here. Here I will write. So these are your T cells, guys. T cells, you have two types of T cells: T helper cells and T cytotoxic cells. These are known as TC cells, guys. Yes. Now T helper cells are also known as CD4 T cells, guys. Yes, CD4 T cells. Your T cytotoxic cells are known as CD8 T cells, guys. CD8 T cells. The main this cell mediated immunity is by your TC cells. TC cells. Now, what are these T helper cells doing then? This T helper cell will activate both your humoral immunity and cell mediated immunity, guys. So, first to come, who will be your Ministry of Defense? Who will be your Ministry of Defense? Your T0 cell. This T0 cell will be your Ministry of Defense, which is the first cell to be activated by your antigen presenting cell in order to proceed with the adaptive immunity further. Now, T0 cells are activated. It has to delineate, or it has to transform into either TH2 cell or TH1 cell. TH2 cell and TH1 cell. So TH is the precursor before the B cell immunity or humoral immunity or TC cell immunity. Cell mediated immunity is taking place. Okay. So the precursor is T helper cell. This T helper cell is also of two types: T helper 2 cell and T helper 1 cell. Yes. Now, so let me come a little from before. This pathogen has entered because your barriers failed, your local immunity failed, the cells and proteins failed, local inflammation failed. It has entered inside. So one of the presenting cell, one of the cell will present it to your Ministry of Defense, that is your T0 cell. Now, this based upon the antigen that is there, if it is intracellular antigen, the T helper cells will be created. So if the antigen that has entered here, if that is an intracellular antigen, the T cells will form T helper 1 cell. If it is an extracellular organism, extracellular means if the organism is there inside the blood, then they will form T helper 2 cell, guys. T helper 2 cell. Now this T helper 1 Suppose this is an intracellular organism. So T helper 1 cells will be activated. Now these T helper 1 cells will activate your TC cells, and this will proceed with your cell mediated immunity. Okay. Now, if the bacteria is extracellular, now let us see what are intracellular and extracellular. TB bacterium, Mycobacterium tuberculosis of yours, is it intracellular or extracellular? It is intracellular, guys, intracellular. So which immunity has to be activated? T helper 1 cell ka immunity has to be, T helper 1 cell has to be activated. Your other bacteria like your Staphylococcus, Streptococcus, your hepatitis viruses, all these are extracellular. That means they are found in the blood. Then which immunity has to be activated? Your B cell immunity or humoral immunity has to be activated. Now, how this will be activated, how T helper cell is activating B cell and how T helper 1 cell is activating TC cells by certain processes that will take place here. Now, these processes we'll be seeing afterwards. So you got a major idea of what is happening. Let us come back to our barriers now. Let us discuss something about innate immunity now. Take a new page. Let us discuss something about what?
थोड़ा बड़ा लिखो.
बड़ा ठीक है. Innate immunity. Okay, I'll write a little bit. Yes, innate immunity. Let us first start. What were the two components of innate immunity? Write down in the chat box. What are the two components that I talked? Yes, may I know in the chat box? Are everyone alert? May I know what is the answer coming? Are they attentive?
Yeah.
Or is my explanation clear?
Barriers. They say.
Barriers and the two things I told you. What are
Barriers
these two
and
things?
local soldiers. Yes.
Local soldiers. What are the local soldiers? Your local inflammation, point number 1. Your cells, point number 2. Your proteins. So we are going to discuss that in detail now. So what are the barriers that we are talking about? Before talking about the barriers, let us talk about the borders that are there. Where should be the barriers? It is not that only from Pakistan side the soldiers will enter, only from China side soldiers will enter. They can enter from the Nepal side, they can enter from Bangladesh side, they can enter from Myanmar, they can enter from Sikkim, they can enter from anywhere. So all the places do we have to have barriers and local immunity? Yes. Now, what are the places in the body from where the organism can enter? First is your skin. The organism can enter if there is a breach in your skin. So first is skin. Yes. Second is your GIT, guys, your GIT. So this is your stomach, this is your duodenum, and let us say this is your anus. Here is the anus, this is the mouth. So they can enter from oral cavity, they can enter from your anal canal. Anal canal. So GIT is the second. So third thing from where they can enter is your respiratory tract. This is your nose, guys, nose. So this is your respiratory tract from they can enter. Fourth thing they can enter from is your eye. They can enter from your eye. And also from along with GIT, they can enter from your urogenital tract from the vagina and the urethra, guys. Yes. So these are the 5 places, mainly 1, 2, 3, 4. So 1, 2, 3, 4, 5 places from where the organisms can enter. From each place, what are the anatomical barriers and physiological barriers? Let us see now. So what is the anatomical barrier we have in the skin? Intactness of skin itself is an anatomical barrier, guys. Intactness of skin. Now, what is maintaining the intactness of this skin, guys? Your keratin, guys. Your keratinization. Keratin is maintaining your intactness. What is secreting this keratin? Your keratinocytes, your corneocytes. Stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, stratum basal. These are your 5 layers. So stratum corneum is your dead layer, which is impervious to water, which is impervious to bacteria. So that is maintaining your intactness of skin. Now, this is the way you have to present in the examination also. Innate immunity is a very, very important answer of yours. So first you have to describe the barriers. Barriers may each place you describe and write the anatomical and physiological barriers. So keratin you have discussed. So these are the 2 things. The second thing in the GIT, what is the anatomical barrier? Your mucous membrane. Mucus. Mucus. So this is the mucous membrane that will be lining. So if the bacteria has entered also, the mucous membrane here, the mucous membrane here will not allow it to invade the enterocytes. Enterocytes means your cells of the intestines. Yes, the mucus. And second important, very important anatomical barrier that is found in your intestines, not in the stomach, but intestine, is your local flora. Your implied thing comes here, local flora. Local flora. What is local flora? So your intestine is full of bacteria, So these are the bacteria. So not even an inch is spared, not even a centimeter is spared. Everywhere there will be bacteria in intestines, basically in small intestines, ileum, jejunum, and your large intestine also. Yes, everywhere there will be local bacteria inhabiting over here. So what is the function of this local bacteria? These are non-pathogenic bacteria, guys. These are non-pathogenic bacteria. Yakult you drink, right? Yakult. What is that Yakult? That is yogurt. So why do we drink that yogurt? Because to increase the local bacteria. What is the local bacteria here? Your lactobacillus. Other bacteria is also there, the local bacteria. If this local bacteria, local flora is gone, this is acting as an anatomical barrier. If this local flora is gone, then there is a space created. Now here is a space created. The local flora is gone. The pathogenic organisms will now occupy this and cause diarrhea and cause diseases. Yes, so local flora is very, very important. If you want to write some clinical aspect over here, write down somewhere else your beside paper that first is your Yakult, that is your probiotics called. Someone has anyone suffered from diarrhea? I am sure all of us have suffered from diarrhea. It is so common in our country. Yes, so diarrhea, what they give? They give your probiotic. What are the probiotics that they give? Yes, yeah, probiotic. Why do they give probiotic? Because the local bacteria should form again. They will inhibit the pathogenic bacteria by certain mechanism. First is by anatomical barrier. Second is by they secrete certain antimicrobial substances also that will destroy the pathogenic bacteria. Yes. Now this is the local flora ka concept. Second applied point that I would like to add over here is that your antibiotics. Indiscriminate use of antibiotics. थोड़ा बुखार हो गया एंटीबायोटिक खा ले रहे, कुछ भी हुआ एंटीबायोटिक खा ले रहे. हाँ, you take antibiotics indiscriminately, what will happen? This local flora will die. What antibiotics? Like your cephalosporins, your aminoglycosides, like your clindamycin and all these antibiotics, if you take indiscriminately, okay, that will cause destruction of this local bacteria. Now the pathogenic bacteria will grow over here and will cause diarrhea. This is known as antibiotic associated diarrhea, or also known as super infection. Because of the death of the local flora, some other flora is growing, so not flora, some other pathogenic organism is growing, that is causing your diarrhea. This is known as your super infection. Very, very, very important history to rule out to if someone is coming with diarrhea. The things what you have to ask: the food history, what he has taken, whether it is a gastroenteritis case, food poisoning or all, food poisoning or something else. The one more important history that you have to take is your antibiotic. Has he taken any antibiotics? Because that may cause super infections. Okay. So for your quick information, remember that super infection is caused by a bacteria that is an anaerobe known as your Clostridium difficile, which causes pseudomembranous enterocolitis. If you want to remember, you remember, otherwise you can leave it. Now, what from the urogenital tract, what are the anatomical barriers? Same, mucus will be there. Okay. Now what is the respiratory tract? What will be the anatomical barriers over here? Your hair in the nasal cavity, hair, they will trap as the anatomical barrier. They are acting. And secondly, mucus will also be there here. Mucus will also be there as an anatomical barrier. Third is your eye. The last is your eye. What will be there? Firstly, the eye is covered. Whenever something is going to enter into the eyelids are going to close. The lids are acting as a mechanical barrier. If the lids are open and the bacteria is entering, your conjunctiva. What is conjunctiva? It is a mucous membrane. So mucous membrane came here also. Conjunctiva will prevent the bacteria from entering, invading the tissue of the eye. Yes, this is what is important over here. Let us now talk. We have talked about the anatomical barriers. Let us now talk about the physiological barriers. What are the physiological barriers? Yes. Yes, excuse me. Why am I using this orange color? I think it's not visible. I'll use some dark color. Yes. Physiological barrier.
Increase the font, ARBAZ.
Okay, sorry, font I will increase the font. Yes, physiological barrier, what is there? Skin ka intactness is anatomical. When will physiological come into play? Whenever the anatomical barrier fails and the organism has entered inside, will the local physiological barrier takes place, come into picture. What is the physiological barrier over here? It is your temperature, guys. It is your temperature. If the temperature increases, the bacteria that has entered into the dermis layer of the skin will get destroyed. Yes, this is the temperature. Temperature acts as a barrier elsewhere also, where it will not allow replication of the bacteria to take place. For replication of the bacteria to take place, there should be adequate optimum temperature required. Yes, if that is not there, this thing will happen. The bacteria will not grow. Here, okay, skin mein ho gaya, anatomical ho gaya, physiological ho gaya. Now GIT, let us see what are the physiological. First is your pH, guys. HCl will be there. Yes, your HCl ka pH. So what is the pH of the gastric juice? May I ask in the chat box. Now, what are the other? Mucous membrane we have talked about, but this mucous membrane will secrete some enzymes. Yes, all these are baby things, you know everything, but I am streamlining everything. Yes. What are the enzymes that they secrete? Enzymes. So first in the mouth you have saliva. In saliva you have lysozyme. Second, when it enters inside the stomach, you have trypsin. Yes. Second, thirdly, if it enters the duodenum and all, you have your bile salts. One of the most important function, guys, of bile salts. Bile salts get 10 functions are there. You will be asked those 10 functions. Patent viva question. GIT, like your entire syllabus will be divided between 4 professors. 2 externals, 2 internals will be there in your viva, and 2 internals will divide some chapters, and 2 externals will divide some chapters. So your entire syllabus will be divided into 4 parts. So one of the professor will take you GIT and some other unit. So if anyone who is taking GIT, this is the patent question. What is the patent question? What are the functions of bile, they will ask. So one of the important function of bile is your antimicrobial property. They will kill. How they will kill? They will destroy the cell wall, they will destroy the cell membrane of the bacteria. Now what is the mechanism of the lysozyme? How they will kill? Let me know in the chat box. Keep telling the answer that they are telling.
Yeah,
or they are not
for your question about the pH of the gastric lumen, they have answered, many of them have answered 1.8.
Okay. Yes, 1 to 1.8. Yes.
We also have other answers, but majority of them said 1.8. Someone
Yes.
said 0.9, 1.7, 6.9, 2.
6.9. Yes.
Yeah.
Then yeah, what is the what I asked next question?
There are no answers for the next question yet.
What I asked, I also forgot.
Where
is lysozyme acting? Where is lysozyme acting? Yes. Where is lysozyme
there's
acting?
an answer. Lysis of cell wall of bacteria, lysozyme action, the cellular lysis, cell wall, mouth, cell wall,
cell wall.
saliva,
NCERT
oral cavity.
Yes, NCERT. Your cell wall of the bacteria is made up of NAG and NAM, N-acetyl glucosamine and N-acetyl buraminic acid. Yes, your NAG and NAM are linked by something known as your glycosidic bond, and 2 NAG and NAM chains are linked by your something known as the side bridge. Okay. Now this lysozyme is going to cleave your NAG and NAM.
Right, it's so minute.
Okay. Okay, I'll write big from now. From now, I'll big. No need to remember this. Just for this thing I told. I think I should not tell all this. Okay, respiratory. What are the physiological things in respiratory? I think there are not many physiological things in the respiratory. Temperature only you can write. Temperature, and I think some antimicrobial substances also released. Okay, what is in the eye? Tear. What are the physiological barriers? Tear. What is there in the tear? Same. What was there in your saliva? Your lysozyme. How it will act? Same. That they will destroy the cell wall of the bacteria.
ARBAZ, there's a question or answer. I don't know. Someone says surfactant in respiratory system.
Excellent. Your respiratory system has your surfactant. What is the surfactant made up of? Your yes, palmitoyl. This thing, lecithin. Dipalmityl phosphatidylcholine, or your lecithin, that is acting as your physiological barrier in the alveoli. Very good. Who told that? Yes.
It's Sayeda. I think.
Yes. So what we have discussed till now what we have that surfactant concept biochemistry mate is very, very important. Your sphingomyelin ratio, your lecithin sphingomyelin, let us not go all that. Okay, this is your barrier. Barrier we have discussed. Now barriers failed. As we are discussing, we will discuss that they failed. Okay, your barrier failed. Now the bacteria has entered inside. So who should come into picture? Local soldiers. What are the local soldiers? Your inflammation cells and proteins. Let me quickly give an introduction about inflammation. Inflammation is a very, very important chapter in your pathology. Yes, immunology is a very, very important unit in your microbiology. Yes, in pathology also immunology is there. In every subject immunology is there. Yes. So let me talk about inflammation. Inflammation. What is the definition of inflammation? Let me ask. Definition. Definition of inflammation. Am I getting any answers? No answers.
No answers
Okay.
yet.
No answers. No one will tell this
Take
answer.
time to find it.
Yes.
Okay,
They
someone
will
says
take time.
response to injury.
Response
Body's
to increase.
response. Response to
Yes. What
injury.
is response? Response to
Body's
injury.
response.
Yes. In the form of
Pallor,
what? In the
color, swelling.
those are your clinical manifestations. Those are your clinical manifestations. Dollar, calor, pallor, rubor, tumor. Those are your clinical manifestations. So inflammation is defined as the Robbins textbook of pathology defines inflammation as it is the sequence of events taking place in a vascularized tissue. In a vascularized tissue, whenever there is an injury in the vascularized tissue, they are the sequence of events taking place in order to bring cells and proteins of immune importance, cells and proteins of immunity to the localized site of injury in order to promote healing. So this was the muscle. So there was an injury. How that injury occurred? Maybe by the entry of the bacteria. Yes. Now what will happen? There will be inflammation. What will inflammation do? They will bring cells and proteins of your immune importance to the localized site so that there is quick recovery. So this is your inflammation. Don't be in a misconception that only bacteria will initiate inside the inflammation. Any form of injury to vascularized tissue will incite inflammation. If my definition was not right, please some panelists write down in the chat box. Yes.
No, it's correct.
You
It's
find
correct.
okay. Yes. The inflammation is there. Inflammation is there. Now what this inflammation is doing, so this is the muscle. So this is the skin. Let us suppose this is the skin. Okay, this is the skin. Injury. This is the injury. Bacteria has come inside. This bacteria will release some substances known as cytokines. These substances known as cytokines. So there are because of these cytokines, there will be some vascular events taking place. And there will be some cells will be there inside this vascular lumen. So there will be some cellular events taking place. Now what are these vascular and cellular events? We need not know. For your understanding, orally I will tell. Vascular events, there will be vasodilation. Because of that, there will be increased blood accumulating over here. Because of that, there will be redness. So one of your sign is rubor. R-U-B-O-R. B-O-R. Rubor. That means redness. Why is there? Because of increased vasodilation and increased blood accumulating over there. Then there will be calor. Calor means you're increased in temperature. Why? Because much blood has accumulated, more metabolic processes going on over there. This is your second point. Third point is tumor. Why is tumor occurring? Tumor means swelling. Why is swelling occurring? Because there will be increased permeability also. As the vasodilation takes place, there will be increased permeability that will cause your tumor. Tumor means swelling. Yes. Rubor, calor, tumor, or dollar. Dollar means your pain. Dollar means your pain. Pain will occur. Why pain will occur? Whenever there is inflammation, there will be some chemical mediators. Your prostaglandins secreted. Prostaglandins mediate pain. What is the prostaglandin that mediate pain? Your PGE2 and PGE2. Mostly PGE2. Prostaglandin is very, very important answer in your biochemistry. If time permits, at the end of the session, if anyone wants to listen about prostaglandins, okay, I will tell a very quick in one page. I will tell how the prostaglandins are synthesized and all. In 15 minutes, I will tell. Okay, dollar. Not related to this topic, related to immunology, but not related to our topic. So I'm not telling now. Rubor, calor, tumor, dollar, and what is one more? Loss of sensation. What is that called? Loss of sensation. So these are the 5 clinical manifestations. Very, very important. It will be asked in your physiology. It will be asked. Viva. This is viva, and what process is causing what, which of these manifestation is important. So loss of sensation over there. So
I
why
think,
this is occurring?
is it loss of sensation?
What is there?
Or impaired sensation.
Impaired sensation. Okay. Otherwise, what is there? You tell. Impaired sensation. Paresthesia, na? Okay. If I'm telling something wrong, please tell me. Yes. Now, what is there? This is the vascular responses. All these occurred due to vascular responses. Now, why the hell these vascular responses are taking place? To bring, if there is vasodilation and if there is entry exit of the fluid, there will be hemo concentration over here. The concentration of blood will decrease. There will be stasis, guys. Stasis. Why do we want stasis? So that all the immune cells that are coming, they should accumulate over here. So the bacteria that is there, they can go outside the lumen and destroy that bacteria. So this is the I don't know why am I getting so much cough. Okay, this is the they should kill the bacteria. So to bring about the
So ARBAZ, the fifth sign is loss of function.
Loss of function. Okay, sorry.
Yeah.
Loss of function. Loss of function over there.
Yeah.
Yes. Okay. Loss of function. What function? Whatever the organs function is there, that function. Don't think that inflammation is going to take place only in the. It will not be an exaggeration to understand any pathology, any disease that you take. In every disease, you will get inflammation. Every disease, you will get inflammation. So inflammation is a very, 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 very important concept that you need to have in mind. So what are the cellular events taking place? That's what I told here. The cells will be there. They have to get stasis over here. Then they have to escape out of this blood vessel and go towards the area where the pathogen is there, so that they can phagocytose. Now, what are the cells? So we talked about we talked about inflammation. One thing is over in the local soldiers. Second is cells. We are talking about now. Cells. I have gone through that KNR-UHS ka prospectus of first year. In the someone sent me, Fawaz, I think he sent me that. In immunity, he has mentioned whatever he has mentioned that I'm teaching. Okay. In immunity, what all should be there that I'm telling. Yes. I think he has mentioned everything only. Nothing is remaining. Yes. So what I was discussing, cells. What are the cells? What I told you, what are the cells? Let me know in the chat box. Please tell in the chat box. What are the cells? Just now we discussed over here. Yes. NEB, neutrophils, eosinophils, and basophils. Sometimes your macrophages also. What is the first line of defense in the cell? Your neutrophils, guys. Your neutrophils will come. Your neutrophils, if they are not able to destroy, then they will release in cytokines, and then macrophages will come. Other cells are also there. Okay. So is it clear? One more cell is NK cells. No need to discuss over here. So these are the cells. Till now, is it clear? Is it clear? Go through this once again. Till now, is it clear? Let us now proceed towards our activation of your immune process. Proteins, I will mention 1-2 points, and then we can proceed to our adaptive immunity.
Yes,
yeah, ask in the
clear.
chat box.
Yeah, clear.
Yes. Yeah. So let us now talk about cells. We have talked about. We'll talk about proteins. What are the proteins? These proteins are known as acute phase reactants, APR. Acute phase. This is not antigen presenting cell, APC, not IPC. Acute phase reactants. What are the proteins? What is their function? These proteins are increased at the time of inflammation. So there will be positive acute phase reactants and negative acute phase reactants. The proteins whose concentration will increase at the time of inflammation are known as positive acute phase reactants, and whose concentration will decrease is known as negative acute phase reactants. So positive acute phase reactants which have antimicrobial property. Antimicrobial property. Yes. So what are those proteins? If I have to tell you, that is just CRP. So any inflammation is taking place, what will doctor order? He will order CRP. In COVID-19 also, what they are ordering? They are ordering CRP, and one more specific acute phase reactants they are ordering is ferritin. Ferritin is also acute phase reactant. What else is ferritin? Is your storage iron. In iron profile also you see, but it is not an a very good indicator of your iron deficiency anemia. Why? Because if there is inflammation, there will be false elevation of your ferritin. That will cause false impression that there is no iron deficiency. So we have to look for other parameters also. So innate immunity, everything we have discussed, right from the barriers, the anatomical, physiological in detail, the area of entry from where all they enter. Okay, what are the functions of each of them in detail? I think you can write 10-page answer with this. Yes, the physiological barriers also I described everything in detail. Then we also discussed about the local soldiers that are there, local inflammation. I gave you an insight about the inflammation. What is it? Yes, they will ask you this. Then I told you about the cells, and then I told you about the proteins. Yes. So
ARBAZ, can you
innate
put
immunity
some light on ciliated epithelium, how the movement of cilia is also important?
Yes. If they want to know about the cilia, ciliated epithelium, so here, apart from the mucus, we also have cilia in the respiratory epithelium. Thanks for informing. Okay, write down cilia over here. The movement of the cilia is also important for your respiratory tract clearance. Okay, the cilia will be beating upwards from the trachea, from the lungs. This thing will go upwards. The bacteria and all that is all entering the mucus as mucus also, and also your dust also. That will enter upwards by the upward movement of the cilia. Where else is ciliar movement important? Ciliary epithelium. Okay, what is the nature of your respiratory epithelium? Let me know in the chat box. What is the nature? Respiratory epithelium. It is pseudostratified ciliated columnar epithelium. Yes, it is ciliated. Why? Because to expel this. This is also one of your anatomical barriers. For one applied, there is one motility disorder of the cilia called Carter-Jenner syndrome. There we find increased respiratory infections due to non-functioning of cilia.
ARBAZ,
And why
did you
do
mention?
okay.
ARBAZ, did
Any
you
doubts they
mention?
can ask now.
Yeah, sweat and sebum in skin.
Sweat and sebum also. Okay, we will add sweat and sebum. Keratin, sweat and sebum. Sweat and sebum. I think that should come in physiological. Okay, sebum. Okay, they will have antimicrobial property and anti-permeability property also. Okay, yes, antimicrobial property is also there. Yes, so this is a complete list now. I think nothing more can be added. If something can be added, you can
Got the ear opening. Ear is also an opening. The cerumen and wax of the ear.
Here also opening. Yes, here also is opening. You can add. There also you have same mucus, ceruminous glands over there. They will secrete sebum, mucus, and all. Intactness of skin is also there inside the external auditory canal, and the tympanic membrane is lined by your epithelia. That also you can write. Okay.
ARBAZ, there's a
But that's
question.
not a major portal of entry. Major portal of entry is yes. What?
There's a
Any
question.
doubts?
Yeah, which immunity is involved in case of malaria parasite?
Wait, we will come. Immunity abhi innate mein hai apana, abhi adaptive mein nahi hai. We will come. We will understand everything.
ARBAZ,
Yes.
would you like to mention about NK cells? Because it is
NK cells
a very
I'm mentioning adaptive immunity. I will mention about adaptive immunity now. Okay.
but that is
Now
an
we
innate
knew.
immunity.
No. It is innate immunity, but it has a major role in adaptive immunity. Yes.
Okay.
I will mention that in adaptive immunity. Yes. That is a cell of innate immunity. Here we should write NK cell. I told you third cell was NK cell, but I was describing it along with your adaptive immunity. Okay. So it is involved both in innate and adaptive immunity. So it is known as innate-like cell. It is innate-like cell, but also involved in your adaptive immunity. Now, these all we will know from inter knowledge, from 10th knowledge, from your MBBS first year knowledge, everything. We knew this, that there will be barriers till anatomical. You got a clarity. But from now we will not know if there is antibody being produced as a part of your humoral immunity, and your TC cells being produced as part of your adaptive cell mediated immunity. This is humoral, and this is your cell mediated. What are the processes taking place for activation of this? I told you over here some process. Where was that? Yes, this activation, antigen presenting cell and all this all. I will discuss this after discussing how the humoral immunity and cell mediated immunity is destroying the bacteria. After that, we will go retrospectively backward and see how the activation is taking place, because then we will be able to understand that better. Okay, we'll do it that way. Now, we will not have much clarity about how this humoral immunity and cell mediated immunity is actually killing. So barriers fail, your local immunity failed, the bacteria has now entered inside your body, it is staying inside the body. What will bacteria do? It will replicate now, replicate and hijack all your body and cause disease. Now the integrated system of this adaptive immunity, this is adaptive immunity we are discussing, adaptive or acquired immunity we are discussing. So that is humoral and cell mediated. Now how this humoral and cell mediated is activated, that is different story. I told I will tell you. Just a second, sorry. Yes. What happened? Yes. Yes. Now, how they are killing? Now, there are 5 processes by which they will kill. 5. 1, 2. Draw like this. 2, 3, 4, 5. Jagah nahi hai. 4, 5. Okay. I will write names over here and separately I will define. These are your 4, 4, 4, 5 marks. 5 marks questions. All these are 5 marks questions. Physiology. Yes. Let us see. Yes. What are the 5 things? The first 4, 1, 2, 3, 4. The first 4 is by your, what is the function of B cell? They will produce antibody. So humoral immunity is also known as your AMI, antibody mediated immunity. Okay, antibody immunity. The 5th one, last one is your cell mediated immunity. So what are the 4 things that an antibody which is produced towards a bacteria? Okay. The immune system was activated, the B cell was now activated, the B cell now produced antibody, okay, against the bacteria. Are you understanding? Hello, let me know in the chat box if you're understanding.
Some say yes, while some others say no.
Okay.
Majority of them say they don't understand. They're not understanding, I mean.
They are understanding?
No, they are not understanding.
They are not.
Someone say they are exhausted. Someone, yeah.
Sorry. Okay. I will come here. So this was your barriers. This was your local immunity. Innate immunity failed. Innate immunity failed. Now, one of the cell of the innate immunity called as the antigen presenting cell will present the antigen to your adaptive immunity ka cell. Adaptive immunity. Now your adaptive immunity is getting activated. Adaptive immunity is getting activated. What is the function of adaptive immunity? Humoral immunity. Humoral immunity is also known as antibody mediated immunity, AMI. That will produce antibody. This is your antibody. One more thing is your TC cells. When will humoral immunity takes place and when will adaptive immunity takes place? Let me know in the chat box. To alert you, I am asking. When will antibody immunity takes place and when will the cell mediated immunity take place? I told you in the starting of the session only.
Adaptive by B cells, humoral extracellular.
Extracellular, intracellular, I'm asking. Whenever the offending agent is intracellular, your TC cell mediated immunity will take place. Whenever this is extracellular, your humoral immunity will take place. Now, how the humoral immunity and cell mediated immunity will kill the bacteria? By 5 processes. 5. 1, 2, 3, 4, 5. 5th process is by cell mediated immunity. First 4 process by humoral immunity. Is it now clear? Type yes or no.
Yeah, they say yes.
Yes. Not even one no should I get? All yes. 100% yes.
Yes, sir.
100%. Yes. 100% yes. Yes. Now these 5 processes let us discuss. Let us write names of these 5 processes here and then we will discuss. So what is the first process is by neutralization. Neutralization. What is happening in neutralization? The antibody. All these are mediated by antibody. Okay. Now what this antibody will do in neutralization? This is your antigen. It will bind to this antigen. Okay. Bind to this antigen. What is antigen actually? This is your bacteria. So a proteinaceous part of your bacteria which is able to incite the immune response is an antigen. Okay. So this antibody will have, okay, structure of antibody, if we discuss, this is your antibody, this is your antibody, a Y-shaped structure. So this is your FAB part, fragment antigen binding. So this part will bind to your antigen, FAB. So this is your FC part, fragment crystallizer. Don't remember that, only remember this is this part, FAB part will bind to your antibody. Structure of antibody is your very important biochemistry question. You know all this. Yes. Now, what is neutralization? The FAB portion of the antibody will bind to the antigen, and now this antigen will now not be able to cause pathogenicity. It has neutralized. Neutralization. What is neutralization? Acid base. They will form salt. So acid is not able to destroy the tissue. So same way, this is the neutralization taking place. Pathogen will now not be able to destroy. Okay, cause pathogenesis. It will not be able to sit on the receptors on the cells and all. So neutralization. Are you clear about neutralization? Are you clear about neutralization? First process by which antibody is preventing disease to occur is by neutralization. Are you clear?
Yes, they understood.
They understood. Second process is by
someone wants to repeat it once again.
Nothing. Neutralization is nothing but antibody will go and sit on the antigen, and this antigen will now not cause any pathogenicity. It has been neutralized. Now this antigen antibody complex that is formed, it will be removed by your kidney, your spleen and all. It will be removed from the body. So bacteria hai andar, but usko jaake ye loga what they are doing, neutralizing. Okay. Next, what is the second thing? Second thing is by your opsonization and phagocytosis. Phagocytosis. What is opsonization and phagocytosis? What is opsonization and phagocytosis? 4 marks, 5 marks, 2 3 marks questions. Opsonization means coating of the antigen. This is an opsonization. This is opson. What is opsonization? Coating of the bacteria with antibody is known as opsonization. Not only antibody, there are some other opsonins also. There are some other opsonins. Whatever is coating the bacteria is known as your opsonin. So antibody is one of the opsonin. Some other opsonin no need to remember is your classical complement pathway. Okay, C3. Your C3 of the complement pathway is your this thing, opsonin. So what is the role of opsonin? Why are we opsonizing? Opsonizing is like marination. Chicken ko marinate karte, we marinate the chicken and vegetables to cook. So it is like making the bacteria tasty, for phagocytosis. Opsonization is always followed by phagocytosis. So this is now neutralized. So this is nothing but, neutralization is nothing but one of opsonization only. Opsonization. If the opsonization is followed by phagocytosis, this is now tasty. Now this FC region of the antibody will react with the, now this is your phagocyte, your macrophage. This will have an FC receptor. So this FC of the antibody will interact with the FC receptor on the macrophage, FC receptor, and this will now get activated. Macrophage will now get activated, and it will form pseudopodia and engulf the bacteria. This is phagocytosis. So what is opsonization? Opsonization is coating of the bacteria with the opsonins such as your antibody in order to enhance the process of phagocytosis to cause preferential engulfment. Are you clear with opsonization and phagocytosis? Write in the chat box.
ARBAZ, there's a doubt. What is the difference between neutralization and opsonization, and how important is the process of opsonization? Can you clear on that?
Yes, opsonization is very important for phagocytosis. Without opsonization also phagocytosis can occur, but opsonization is enhancing the process of phagocytosis very this thing. If there is chicken placed like that, and if there is marinated and cooked chicken, which will you take? You will always take marinated and cooked thing. Why? Because that is preferred by you. So opsonization is very important for your phagocytosis to enhance the process of phagocytosis. What is neutralization and what is the difference between opsonization and neutralization? After opsonization will the antigen be neutralized, but to remove this after neutralization it has to undergo phagocytosis. I hope this is clear.
Yeah,
is it becoming
one thing? Opsonization is nothing but making the antigen or the bacteria or whatever microorganism, making it tasty. when the opsonins
Yes.
are coated, that becomes tasty, and phagocyte, it loves to eat tasty things. So we increase the efficiency of phagocytosis does.
Yes, yes,
And
it is making it tastier,
for
preferential
basically,
because of marrying. Yes.
it is for soluble. I think it's for soluble. Then the
वो
antibodies
सब
will
नको,
attack
soluble
that.
वो नको नको, इलों को नको,
Okay.
soluble voluble. चलो.
The antibodies will attack that antigen. खत्म, वहीं खत्म, उसको phagocytosis की जरूरत नहीं है, evacuate हो जाता neutralization से, वहां पे उसका end हो जाता.
Yes.
But here in opsonization, you are making it tasty. पूरा उसकी infectivity नहीं खत्म हो रही. The macrophage should come and engulf. तो जब खत्म होता है उसका पूरा game over.
Yes.
Okay, ऐसा है
Yes.
difference.
Yes. Okay. Thanks for adding. I hope you got that. Neutralization is basically neutralizing the bacteria, but it is not completely removing this. Yes. So if there is phagocytosis, this will completely be destroyed. So that is the reason neutralized bacteria should undergo phagocytosis. Yes. Now, what is there? Third thing is by complement mediated lysis. So how the humoral immunity is killing the bacteria, is antibodies killing the bacteria, is by these 4 processes. First two we discussed. The first is neutralization. Second is opsonization, phagocytosis. Phagocytosis, some more detail I will describe after the session. I will describe. Okay. What is complement mediated lysis? Complement. What is complement? Let me tell what is complement system. Yes. Do I have any answers?
No answers yet. Someone says suggesting.
अच्छा.
I don't know what does that mean. Helping. Additional help.
Additional
Yes.
help.
Additional help. Yes. What additional help? Okay. Let me give you
Cascade of
clarity what
proteins.
this complement system is. वो फर्स्ट ईयर में complement system बोले तो हैबत होती थी मेरे को, क्या था कि ये इतना बड़े processes रहते हैं. एक second. I'll take one paper. So this is the third process that we are discussing. What is this? 4-5 marks question, short answer question. Complement system. Complement. I'll not describe in detail what complement system is, not required for first year, but basically complement basically are a group of proteins. Proteins present in the blood, produced by liver. Some are produced by spleen, some are produced by intestines also. Okay, wherever they are produced, we are not bothering. So what are they? They are a group of proteins. What they will do? They will augment the immune response. That means they are helping in killing the bacteria. So these are the 4 processes by which the bacteria get killed. So one of the processes by complement system. So they are helping in killing the bacteria or augmenting the immune response. In complex language, you can say augmenting the immune response or increasing the immune response. So they are nothing but proteins. Now, how these proteins are getting activated? There are three pathways. Class, remember, mnemonic is class. CLASS. Khalil sir gave this mnemonic, class. CLA. C means classical, L means lectin, and A means alternate. So classical pathway, 3 pathways are there. Classical pathway, lectin pathway, mannose binding lectin bolte. So lectin pathway, and alternate pathway. Yes, these are the 3 pathways. Only classical pathway is activated by your antibody. Now, how will then lectin and alternate pathway get activated? These are getting activated directly by the microbe. No need of antibody. So these will come under adaptive immunity or innate immunity. Antibody is not needed, only bacteria is needed. Innate immunity or adaptive immunity. Let me see in the chat box. What are you telling?
Innate this here.
Lectin and alternate pathway. Let us now write down one more thing over here. Cells में proteins and acute phase reactants we wrote, right? So one more thing, you add 2 pathways, your L and A. Your lectin and alternate will be of this, okay? Pathways of your innate immunity. So what is this doing actually? What is this doing? We'll be completing in, I think, more 15 minutes. Over. Only these 5 processes we have to discuss, and something I will tell about the activation, and then we are good to go. I'll revise and then we can disperse. Yes. Now, all these pathways, different pathways will now converge to one common pathway, which will eventually lead to the formation of something known as MAC. What is MAC? Membrane attack complex. Membrane attack complex. Now, this membrane attack complex, what it will do, it will go, if this is the cell of the bacteria, it will attack the membrane and form holes in the membrane of the bacteria. This will cause entry of the fluid inside. This will cause swelling and burst, swelling and killing of the bacteria. So eventually we have to kill the bacteria. So it is killing the bacteria by MAC, membrane attack complex. Only your classical complement pathway. How will you remember only classical? C for classical, C for complex. Complex means antigen antibody complex. So antigen antibody complex is initiating your okay. How it is initiating? Let me tell you. If this is the bacteria, if this is the antibody, this is the opsonized bacteria. Antibody is coded. So this is the FC portion. Now one of the protein of the classical pathway will bind over here, and then it will activate the classical pathway. So FC receptor of your antibody is activating your classical pathway. The same FC receptor is activating your phagocytosis also. Okay. So what is complement system? They are nothing but a group of proteins which will at the end form membrane attack complex, which will destroy the cell of the bacteria. So how is this membrane attack complex getting activated? By three processes: classical pathway, lectin pathway, and alternate pathway. Okay, is it clear?
Arbaz, can you please repeat the complement system again? I mean the MAC part.
MAC part, they want to repeat. So guys, complements are group of proteins, right? Now these proteins what they will do, they are sequence of reactions, cascade. Your coagulation cascade रहता ना, 12, 9, 11, 13 वैसा intrinsic, extrinsic, same वैसी यहाँ पे भी, here also you will have a sequence of processes taking place. At the end of the sequence of processes, there will be membrane attack complex. A structure will be formed that is known as membrane attack complex. It is like a pore. Now this pore will go and sit on the membrane. It will cause pore on the membrane. Now water will inside in the membrane, come inside the cell of the bacteria. This is the bacteria, and will cause lysis. So complement is causing lysis of the bacteria by formation of membrane attack complex. For this membrane attack complex to be formed, there should be activation of this membrane attack complex. So there are three processes: classical, lectin, and alternate. These two are the innate immunity. These two comes under innate immunity. And classical is initiated by your FC portion of the antibody, which is this is thing. Okay. Antigen antibody complex. So someone will say antibodies are freely found in the blood also. Will the FC region of the free antibody also activate the classical pathway? No, only an opsonized bacteria and the antibody which is bound to the antigen. To say antigen antibody complex only can activate the classical pathway. Hope I am clear with the complement system. This is all which is needed. Here they will come C1, CQR, CQS, C2. Okay. Here they will come C3, C5, C6, C7. Membrane attack complex, you remember it is C5 to C9. If you don't want to remember, don't remember. These are the sequence of proteins that are taking. Okay. Don't remember. Is it okay? Is it becoming exhaustive? If it is becoming exhaustive, no need to worry. We are anyways going to end it in more 10 minutes. Everything is over. Only two things are there. Now will come three things are over. 1, 2, 3. Okay. First was your neutralization. Second was your opsonization and phagocytosis. Third was your complement activation. Fourth is your NK cell mediated lysis. NK cell mediated lysis. Now, how are the NK cell getting activated? NK cell, like your complement pathway, the one of the pathway was adaptive and two other pathways were innate immunity. Same here, NK cell also, one of the pathway by which it gets activated is your adaptive, and two other pathways are by your innate immunity. So these two cells are overlapping, not cells, these complement system and NK cells are overlapping in between your innate also and your adaptive also. Hope you are getting what I'm saying. Immunology is a complex topic to understand, but I'm trying to explain you in simple words. Yes. So what I was discussing, NK cell. Okay, NK cell I was discussing. Now this is NK cell. What is NK cell? Let me give you a quick description. NEBLM tha, NEBLM. So these were your lymphocytes. Lymphocytes, you have B lymphocytes, T lymphocytes. One more lymphocyte from now you remember is your NK cell. This is known as large granular lymphocyte. So NK cell is a lineage of your lymphocyte only. Viva question they will ask. Hematology mein you will have the differential leukocyte count. Yes, at that time they will ask many questions from immunology also. They will ask in Grand Viva also. Yes. Did you understand what is NK cell? It is one of the lymphocyte, large granular lymphocyte it is called. Yes. Now what are the markers and all that we will see in microbiology, no need to remember. Now how this NK cell is killing the bacteria? Our ultimate aim is to kill the bacteria by whatever means, whether it be by neutralization, whether it be by your phagocytosis and oxonization, whether it be by complement mediated lysis. One more is by your NK cell. What it will do, same antigen antibody complex is activating your this thing also. Here, this is your FC receptor, same like phagocyte. This is your FC receptor, and this is your FC component of your antibody. So this will now activate. Now, phagocyte was causing forming pseudopodia and engulfing. This is not phagocytosis. Now, what it will do? It will release perforins and granzymes. Perforins and granzymes. Now, this will kill the bacteria. Now, same process is taking place in your fifth thing. What is your fifth thing? NK cell we discussed. Fifth thing is your cytotoxicity. This is the only pathway by which your cell-mediated immunity will kill the bacteria. Yeah, cytotoxicity. Mediated by your which cell? TC cells. Yes, TC cells. Now, same here. What will happen? Antibody ka no role over here. So this is the TC cell. Okay, this is the cell in which the bacteria is there. So this is activated only in intracellular cell mediated. So the bacteria is inside the cell. Now what this will do? This will destroy the entire cell only. Bomb blast kar do, suicide bombing. Okay, what it will do? The entire house in which the bacteria is residing, it will destroy the entire house by releasing perforins and granzymes. Perforins will cause pores, granzymes will enter, and they will activate that gas spaces and all, and will cause apoptosis. Okay. Now, how this will activate? What is the activation process? Here, antibody was there. What is there over here? Here is something known as your MHC1 molecule. It will react with MHC1 molecule. What is MHC? MHC we will discuss now. So, are all these 5 processes by which your adaptive or acquired immunity kills the bacteria? Is it clear? I will do a quick recap from behind. We will come retrospectively. We will meanwhile discuss the this thing also, how the activation is taking place. Then I will quickly revise this. Then we will end the session in another 5 to 10 minutes. Is it clear? Do you want anything in clarity over here? Anything else?
Fourth and fifth repeat.
Fourth and fifth repeat. And fifth repeat. Okay. Fourth is by NK cell. This is known as ADCC. Okay. Antibody dependent cell mediated cytotoxicity. Okay. Yes. Now fifth process is your cytotoxicity. The cell in which the bacteria is residing, the cell itself you are killing. Who is killing? TC cells. Is this your B cell mediated humoral immunity or adaptive immunity? This is your adaptive immunity. Okay. What it will do? It will recognize the cell in which the bacteria is residing with the help of MHC1. Now this what is MHC1 doing? It is presenting one of the bacteria. Okay, this is acting like an antigen presenting cell over here, but no, don't name it as antigen presenting cell. Don't name it. Okay, antigen presenting cell is something different. It will have MHC2. MHC we are going to discuss anyhow. So this is your TC cell. It will recognize. It will know that bacteria is sitting inside. Okay, so now it will release perforins, granzymes. These are your bombs. It will bomb this cell. It will kill the cell by making pores, perforins by pores, and granzymes will enter and kill the bacteria, kill the cell only, entire cell in which the bacteria, along with it, bacteria will also get killed. What is NK cell? NK cell is one of the lineage of a lymphocyte. Large granular lymphocyte is called. As your B cell, T cell is there, one of your cell lymphocyte is your NK cell. What it will do? It will recognize the antigen antibody complex. FC receptor it is having, so it will recognize the FC portion of the antibody, and now it will release perforins and granzymes similar like this, and some other proteins also it will release. And it will cause lysis of only the bacteria. Okay, this is your antibody dependent cell mediated cytotoxicity. ADCC it is called. What are the other 2 processes by which it will kill? It that comes in your innate immunity. I told you no, we are not discussing. At this level. It is very, very exhaustive to discuss. Hope this is clear. All these 5 processes are clear. Let us now proceed to this thing. One more thing. I hope I've discussed almost everything. Some more things I will just brush up upon. So these are, suppose you have undergone an injury. The skin is breached. So your barrier is gone. So bacteria has entered inside. So here has to be your innate cells, innate immunity cells. What were the innate immunity cells? Neutrophils and macrophages. So first neutrophil will come, it will try to engulf. It is not able to kill. Okay, what it will do, it will release some cytokines. What are cytokines? There are certain chemical messengers. So chemical messengers. It will call macrophage. Macrophage will come. It will try to kill. It is also not able to kill. It is also not able to kill. So what will do? The bacteria is coming, but innate immunity cells and proteins are not able to kill. So proteins are local soldiers also failed. Okay, inflammation failed. Inflammation has brought these things over here, that also failed. So it failed. So now what we will do, one of the cell called as antigen presenting cell, it will engulf this bacteria. Now it has engulfed. Now what it will do, it will break down this bacteria, and present one of the fragment of this bacteria via MHC2, major histocompatibility complex 2, to your central government. What is it? T0. So what is antigen presenting cell? If you want to write down, beside you write down, this is a very important VSQ, very short answer question. Okay, now antigen presenting cell is a cell which is mediating your initiating your adaptive immunity. Now from here innate immunity failed, from here adaptive immunity is going to occur. In adaptive immunity, what will happen? Antigen presenting cell will present the cell to T0 cell. So antigen presenting cells are the cells which will engulf the bacteria, process the bacteria, break down the bacteria into fragments, and present one of the fragment of the bacteria via MHC molecule to the T cell, Okay, I hope antigen presenting cell ka definition is clear. Now what are the different types of antigen presenting cell? You remember only 3 types. Most important antigen presenting cell is your dendritic cell. What is dendritic cell? Dendritic cell is a type of antigen presenting cell. Where it is found everywhere. Majorly it is found in skin. Your stratum lucidum, I think, your stratum granulosum, I don't know. Somewhere in the skin it is found. Okay, whenever there is bacterial entry, this antigen presenting cell will engulf and immediately present to this thing. When it will present, when the innate immunity is failing, it will present. Yes. Now, second is your macrophage. Macrophage is also one of the antigen presenting cell. Third is your B cell. So these 3 cells can act as antigen presenting cell. Hope you are clear with what is antigen presenting cell. What is MHC? MHC is nothing but there are receptors. There are receptors which are helping in presenting the antigen. So MHC2 cells are found on antigen presenting cell. And MHC1 cell we have discussed over here, where we discussed MHC1. Over here we discussed MHC1. MHC1 is found in all nucleated cells. So whenever all nucleated cells are infested by the bacteria, so they will also process, fragment and all, they will also present to TC cells. So MHC1 is presenting to TC cells, and MHC2 is presenting to your T0 cell, T helper cell also. Okay. Now T0 cell will now what they will T helper cell, T helper 1 cell, T helper 1 cell, or T helper 2 cell, TH1 cell or TH2 cell. When will T helper 1 cell form? When will T helper 2 cell form? When intracellular bacteria is there, T helper 1 cell is formed. Okay, that will mediate cytotoxic immunity, cell-mediated immunity. Whenever there is extracellular bacteria in the blood or virus, then it will cause your humoral immunity. Humoral immunity. What is there? Your B cell is there. B cell is activated. How is B cell activated? No need to remember this, that there is a somatic hypermutation, affinity maturation, and all that will take place. But just remember that T helper 2 cell is activating a B cell. Now B cell will activate to plasma cell. This is the plasma cell. Okay, this is the plasma cell. Okay, now this plasma cell will secrete antibodies. Now what will this antibody do? This antibody will do these 4 processes. 1, 2, 3, 4. 1, 2, 3, 4. Now same way here, T helper 1 cell, by releasing certain cytokines, interleukin 2, interferon gamma and all, they will activate your TC cell. Along with that, they will activate macrophages and all. No need to remember, TC cell will activate. What this TC cell will do now? This TC cell will do cytotoxic immunity. 5th process. What is that cytotoxic immunity? I already discussed. Cytotoxic immunity is nothing but they will recognize the bacteria with the help of MHC1. All nucleated cell it is present. So if the bacteria is present inside, or virus is present inside, or tumor cell is present inside, or your virus is present inside, they will recognize it and release perforins and granzymes and kill the bacteria, kill the cell in which the bacteria is there. Hope this is clear. Hope this is very, very clear. Yes. What else I have to discuss?
ARBAZ, someone asked, what is second type of APC?
Second type, macrophage.
Okay.
Macrophage. APC will have MHC class 2. So if they want to write definition of MHC class 2, complicated definition is given in textbooks. MHC class 2 is nothing but they are proteinaceous receptor present on cells. MHC1 is present on your all nucleated cells. and platelets, and MHC2 is present on your antigen presenting cell. What they are doing, they are nothing but they are presenting the antigen, one of the peptide of antigen, which is fragmented by the antigen presenting cell to the cells of immunity. Your T0 cell or T helper cell, yes, they will present. Okay, that is the thing. Same
ARBAZ,
applies to T helper
also discussing? Are you also discussing toll-like receptors
You're not,
today? Toll-like receptors.
You're not audible. Repeat, please. Yes.
Can you hear me now?
Yeah.
Toll-like receptors. Someone saying
they
that
want toll-like receptor also. Okay.
Yeah, someone saying that toll-like receptors were also there in the prospectus.
Okay, toll-like receptor also there in prospectus. Let us discuss toll-like receptor also. Nothing. So what is there?
And ARBAZ,
Here
they also want prostaglandins to be discussed in the end.
Prostaglandins you want? Okay, we will discuss prostaglandins also. Okay. So here the bacteria was entering inside. Okay. First the bacteria was entering inside. Okay, this is getting bad. I think we are done with the, okay. Am I, hello?
Yeah, your screen is blurred.
It is getting so much blurred.
Extremely blurred.
Yes, I'm seeing.
Better.
It's okay now.
Okay.
Write a
So
bit bigger.
other people, if you, I think we are done with the class. We are done with the session about immunity. I have also revised the immunity. Okay. I will revise some more. Okay. Other people, what doubts they are asking, if you want to listen, you can stay. Otherwise, you can leave the session. Okay. So what they were asking, toll-like receptor, TLR. So here, what happened? Your bacteria, skin was breached, your bacteria entered inside. Your cells of innate immunity, like your neutrophils and macrophages, should recognize this bacteria. Now, how are they going to recognize? This is not coated. Like your B cell and T cell, how they are recognizing? As it is coated by your antibody, they are recognizing. And TC cell, how it is recognizing? By your MHC class 1, it is recognizing. But these innate immunity cells, they have nothing to recognize. How they will recognize? There is no antibody, there is no MHC. So they will recognize something known as your PAMPS. So this is your bacteria. There will be some sequences. Known as PAMPS. What is PAMPS? Pathogen associated molecular patterns. So if this pathogen associated molecular pattern is present on the bacteria, they will recognize. Who will recognize? The neutrophil. Your neutrophil will recognize. With the help of what? With the help of one receptor. This receptor is known as your toll-like receptor T. This is known as your toll-like receptor. Is it clear what toll-like receptor and pathogen associated molecular pattern is? This is TLR. And also we have DAMPS, DAMPS, disease associated molecular patterns also we have. Here, if the cell is undergoing any disease, then also it has to be apoptosized by the cells. So they will be having disease associated molecular patterns. Your bacteria will be having pathogen associated molecular patterns. And your cells of immunity will have receptors, your toll-like receptor, which will recognize this pathogen associated molecular patterns. Okay. And it will engulf what all it has to do. It will undergo. It will phagocytose, or it will kill the bacteria. Okay. Now, what are the cells with phagocytic property? Can I know what all cells has phagocytic property in the body? Now, this toll-like receptor, some other receptor will also be there. Okay, mannose binding lectin receptors, and your other receptor I'm not remembering. What are they? All these are known as your pattern recognition receptor. I hope this is clear. TLR is one of the pattern. It is recognizing the pattern. It is one of the pattern recognition receptor. It is TLR. Okay. Yes. Now, what else they had the doubt? Prostaglandins they want. I will discuss prostaglandins very quickly. I will discuss. Is it clear? Toll-like receptor. Who asked?
You just asked to name the phagocytes. We have an answer.
Are they naming?
Someone said Kupffer cells and glial cells.
Aha! You don't know the concept of phagocytosis only. Phagocytes are by your blood cells. What are the blood cells? ये गज़ाब है स्किन का. neutrophil, eosinophil, NEBLM na? Yes. Your neutrophil, your basophil, and your monocyte. Monocyte are your phagocytes. This monocyte will go into certain tissues. Now, when it is going to tissue, it is known as your macrophage. Macrophage. When it is going to your bone, what it is known as? Osteoclast. Osteoclast. When it is going to your placenta, it is known as your half-Bower cells. Half-Bower cells. When it is going to your brain, it is called not glial cell. Don't say glial cells. All glial cells are not phagocytosing. Your oligodendrocytes are not phagocytosing. Only your microglia. What is the derivative of microglia? It is ectodermal or mesodermal in origin. Mesodermal. Why? It is nothing but your blood cell. Blood cells are mesodermal in origin. Very, very important. Yes. What else are there in your liver? It is known as your Kupffer cells. Kupffer cells. So these are all your phagocytic cells. You want phagocytosis in more detail? I can tell, but I don't think it is necessary. Let me now discuss about the prostaglandins. Very, very important. Yes.
Tissue basophils.
Yes, tissue basophils also I told.
Huh?
Neutrophils. I told, huh?
What are basophils called when they go to the tissue?
Okay, they are called mast cells. They are called mast cells. They are called mast cells. They are also phagocytes. Okay, they are not primary phagocytic cells are macrophages. Primary phagocytic cells are your monocytes. Your macrophages are primary phagocytes. Now, what else do we have to discuss? Prostaglandins. I did not revise prostaglandins, but I will tell what all I remember. I will tell. Prostaglandins. Prostaglandins. Okay. What are prostaglandins? These are certain chemicals. These are certain chemicals. They are helping in your inflammation, and your immunity. And they will have other functions. What are the other functions? We'll discuss with each prostaglandin. So what is the inflammation? Why they are causing inflammation? So their what in inflammation? Your vasodilation. Some mediators should be there. Your increased vascular permeability. Here also something should be there. So these two things are being caused by your prostaglandins. In immunity, what are they causing? They are your chemokines. Chemotaxis they are causing. Chemotaxis. Okay. Main is in vasodilation, and one more is pain. So all the process I told you, dollar, calar, pallor, rubor, and tumor, all those were there, right? So why am I telling pallor? Pallor will not be there. Dollar, calar, rubor, tumor, and loss of function. Yes, all these are mediated by your prostaglandins only. Your vasodilation by prostaglandins, your vascular permeability by prostaglandins, your calar or pain by your prostaglandins, chemotaxis by prostaglandins, and also your other things are also there. Other functions we'll discuss in other functions. What are the other functions in GIT? In GIT,
ARBAZ,
they will decrease acid production. They will decrease acid production and increase mucus production. So other functions one function is they will decrease acid production and increase mucus production. So they are protective, right? Protective for your GIT. So painkillers you take. What are painkillers doing? Screen is black.
Yeah, clear now.
Clear. Yes. What are painkillers doing, guys? Painkillers are decreasing this pain. Now, this pain, guys, this pain is mediated by prostaglandins. So to decrease this pain, you have to decrease prostaglandin synthesis. What are your painkillers? Let me know in the chat box. MBBS first year is getting over. At least you should be able to prescribe painkillers to somebody. Let me know what are your painkillers. What are they known as? Which classification do they come in? Pharmacological classification.
Yeah, someone said Nsaids.
Nsaids. What are Nsaids? Non-steroidal anti-inflammatory drugs. What are those? We are going to discuss very detail. Yes. What are Nsaids? Don't tell this the class name. Tell the drug name also. Don't tell Dolo. Don't tell brand names. Tell the generic name.
Yeah, someone said paracetamol.
Paracetamol.
Yeah,
and
paracetamol.
Paracetamol. Any layperson will also know. Everyone will know paracetamol.
Someone said Lyser D. Aspirin. Yes.
Yes, aspirin. And what else? Yes, and what else?
Someone said Lyser D, Fipronil, ibuprofen, aceclofenac. What else do we have?
Yes, you're doing it. Very good. You're
Diclofenac.
doing all those things here. Yes.
Diclofenac.
Diclofenac, aceclofenac, temozolide,
Uh,
etodolac, meloxicam,
what this?
aspirin, diflunisal. These are all your Nsaids, painkillers. What they will do? They will decrease the synthesis of prostaglandins and decrease the pain. Now, pathways of prostaglandins, let us discuss. Pathway, how this is synthesized, how prostaglandins are synthesized. This is your cell membrane. Cell membrane will have phospholipids, lipids. Now, these phospholipids are acted upon by your phospholipase C, to form arachidonic acid. Arachidonic acid. Yes, phospholipase C, arachidonic acid. Now, this arachidonic acid, what is this? It is a derivative of your phospholipid. Now, this arachidonic acid will now be forming your what they will form, Pranav? Maybe Shanbag name will be. I did not revise, so I'm just getting.
2 pathways आते हैं ना,
somewhat
arachidonic में lipoxygenase, cyclooxygenase.
Yeah. Okay, one second. कहाँ गया वो NSAIDs का? Yeah. Are you able to see this over here? Okay, I will write down only. So phospholipase A2 है ये. Phospholipase A2 will form arachidonic acid. So this now arachidonic acid will now be acted by two, as Nusrat told, 2 enzymes. Okay, if it is acted by your LOX, it will form leukotrienes. If it is acting by your COX, it will form prostaglandins. Okay, this is your COX, this is your LOX. Okay, now this your prostaglandins, your Nsaids are inhibiting your COX. Nsaids are inhibiting your COX. Nsaids. So painkillers are inhibiting COX, so that prostaglandins should not be synthesized, so that they will not cause any symptoms. Now, what are the prostaglandins? PGE2. 3 prostaglandins you remember: PGE2, PGI2, and PGF2 alpha. What will PGE2 do? PGE2. Now, they are classified based on the site where they are present. If they are present in the tissues, that is known as PGE2, and they will cause pain. And if they are present in the GIT, what I told they will do? They will decrease the acid production and increase the mucus production. So they are protective for stomach. If you know, whenever we give steroids are inhibiting prostaglandins, phospholipase A2. Okay, it will also decrease pain. If there is severe pain, we give steroids, but it will also decrease LOX pathway and leukotriene synthesis also. Okay, now what I told you that HCl and mucus is HCl is decreasing and mucus is increasing. That means PGE2 is protective for stomach. So whenever anyone is suffering from Bell's palsy or any disease where they required steroids or where they required high dose of NSAIDs, what doctor will give along with painkiller? Can anybody tell me? What he will give? He will give Pandy. What is Pandy? Anyone know? Pan 40, Pandy, pantoprazole, omeprazole, lansoprazole. What are all these?
Said antacids.
Okay, these are not antacids. These are decreasing your acid, but these are not antacids.
Yeah, they answered. Someone answered proton pump blocker.
Yeah, these are proton pump inhibitors. Your PPIs. Why they will decrease? Why we are giving PPIs along with this? Because this protective mechanism is lost. If we give steroids and NSAIDs, this protective mechanism is lost. So now HCl will be increased, mucus will be decreased. That will destroy the stomach. That will cause ulcer. That will cause. So along with this, we give Pandy also. Pandy or Pan 40. Is it clear? Applied aspect. So Pandy or Pan 40, pantoprazole is your proton pump inhibitor. Do you now know why we give Pandy or Pan 40 along with NSAIDs? Okay, along with leukotriene, montelukast also we give. Yes. Now, what will happen now? PGI2. What will happen in PGI2? PGI2 is also known as prostacyclin. And one more thing is TXA2. This TXA2 and PGI2 will have opposite function.
These are present in platelets. Where were these present? If it is present in tissue, PGI2 present in tissue, pain. If it is present in GIT, these things. Now these are present in platelets, and one second. PGI2 is present in your endothelium and platelets, and thromboxane A2 is present in platelets. So what they will do? They will cause thromboxane A2 will cause platelet aggregation. Platelet aggregation. It is helping in coagulation pathway and your vasoconstriction also. Opposite you write for prostacyclin. So you will write, what you will write? Platelet aggregation ka inhibition of platelet aggregation, and vasodilation. So which is protective for human being? PGI2 or 3xA2, which is protective in MI, myocardial infarction, which is protective. In 5 minutes we'll discuss this and we'll close off this.
I2
Yes.
They say.
I2 is protective. I2 is protective for human body. Now, what aspirin? You know aspirin? Aspirin is also one of the NSAID, but it is an irreversible inhibitor of your COX pathway. Now, what is aspirin? Is the most widely used drug in the body, in the world. Now, for what indication is aspirin used? Can anyone tell? Even lay person will know this. Aspirin is never ever used for pain, guys, because it is irreversible inhibitor. It will cause ulcer, stomach, bleeding, perforation, death. Don't use for
Yes,
pain.
they answered.
Then for what?
Yes, what? Yeah, they answered myocardial infarction. Someone
yes.
asked, is it clot buster?
Not clot buster. It is not clot buster. Clot buster means thrombolysis, it is called. Those are your thrombolysis, your other things. These are your antiplatelets. Platelet aggregation, it is inhibiting. So in coagulation, you have first platelet aggregation, then on that your clot formation will take place, right? So clot formation is a different thing. So clot lysis is a different thing. Okay, thrombolysis it is called. TPA, tissue plasminogen activator, those drugs are called. Here platelet aggregation they are inhibiting. So these are inhibiting. Aspirin is inhibiting low dose aspirin. Low dose aspirin is inhibiting your platelet aggregation by increasing prostacyclin and decreasing thromboxane A2. Increasing prostaglandins and decreasing prostacyclin and decreasing. Now you will tell both are synthesized by COX pathway. If aspirin is inhibiting the COX pathway, one is increasing, one is decreasing. How it can happen like that? There is some trick over here. If you want me to tell, I will tell, but it is not required actually. Okay. Basically, these are present in your platelets, right? Your thromboxane A2 is present in your platelets, and endothelium is present in your endothelium. So endothelium is nucleated and platelet is not nucleated. It is irreversibly inhibiting. So after irreversible inhibition, if the nucleus is there, they will synthesize new COX. So nucleus is there in whom? In endothelial cells is there. So prostacyclin new can be synthesized. So prostacyclin can be is increasing, and thromboxane A2, as nucleus is not there in platelets, they will decrease. I hope this is clear. Now PGF2 alpha, last thing in prostaglandin is PGF2 alpha. They are causing uterine contraction. This is your uterotonic drug. One of your uterotonic drug. It is causing uterine contraction. Hope this is clear. All these things are clear. How phospholipids, which enzyme is acting, what is arachidonic acid, structure of arachidonic also you remember in biochemistry. What is COX? Cyclooxygenase. 1 and 2 we have. Okay. So remember there are two cyclooxygenase, 1 and 2. So Nsaids will be there, and aspirin is irreversibly inhibiting the COX. Because of it is irreversibly inhibiting, we are utilizing it in MI, myocardial infarction. And what are the other things? Main important is pain. We give for pain. PGE2 is mediating pain and also mediating your HCl, decreased HCl and increased mucus. Protective for your GIT, and also your PGF2 alpha. What it is doing? Uterine contraction. In parturition it is helping. Your parturition it is helping. Other vasodilation it is helping, also vasodilation. So it is also giving impotence and all it is given. So this is all in nutshell about the prostaglandins. Okay. I hope this all is clear. Yeah. If you want anything else, you can tell. Otherwise, we will end the session.
Yeah, explain PAND function once.
PAND function they want. Okay. PAND is nothing but your paracetamol. What is paracetamol? It is an NSAID. What will NSAID do? NSAID will decrease this COX, inhibit this COX, reversibly inhibit the COX. If there is inhibition of COX, what will happen? Prostaglandin will not be synthesized. So your PGE2 is not synthesized. PGE2 is very, very important for pain mediation. Your pain will not be mediated now. Pain will not be mediated. So decrease pain. It is an analgesic, guys. It is an analgesic. So to decrease pain, it is causing this thing. And this is also cause vasodilation and all. It is anti-inflammatory also. No, paracetamol is not anti-inflammatory. It is analgesic and antipyretic. Okay, one more important thing I forgot to you, fever. PGE2 is also causing fever. It will go to your hypothalamus, anterior hypothalamus it will go and increase the set point. Why it will cause fever? It is one of the protective mechanism, right, in the body. So it will also increase the set point in the hypothalamus and it will cause fever. Hope that is clear. So to decrease the fever also we give Nsaids, paracetamol. So it is decreasing pain and decreasing fever also. Yes, it is antipyretic and analgesic. Other anti-inflammatory drugs, Nsaids are anti-inflammatory also, but paracetamol is not anti-inflammatory. It is only analgesic and antipyretic. Anything else? Hope that was clear. If it was not clear, let me know. I will repeat. Yes. Immunology topic, if anything is not clear, in prospectus anything else is given, and if you want to know, I will tell. I don't want it to overwhelm for other students who don't want to listen, so I just wrapped up it. Yes.
Yeah, PGI2 once, please.
PGI2 they want? PGI2. Okay. What is PGI2? You have asked. We have all these prostaglandins, PGE2, PG thromboxane E2, PGI2 and PGF2 alpha, all these are there. So there are two things, thromboxane E2 and prostacyclins, PGI2. This is called prostacyclin. Now where are these present? Thromboxane E2 is present in your platelets, and PGI2 is present in your endothelium. Now what are their functions? Thromboxane E2 is causing platelet aggregation, and your vasoconstriction. Opposite functions PGI2 is causing platelet aggregation, inhibition of platelet aggregation, and vasodilation. So in body there is balance between two. There is ratio is maintained between these two, thromboxane A2 and PGI2. If TXA2 is increasing, then there will be MI. Why? There will be clot formation. If stroke also will be there. If there is increase in PGI2, there will be bleeding because clot is not forming. Platelet aggregation is not occurring. So to enhance the to decrease the platelet aggregation, we want to increase the PGI2. So we give low dose aspirin. It will increase the PGI2 and decrease the thromboxane A2. Okay, I think that was clear. Leukotrienes I did not mention. I will mention about the leukotrienes also. I will mention. For arachidonic acid, they can form from LOX pathway, lipooxygenase pathway. They can form. This is an enzyme, lipoxygenase. They will form leukotrienes also. Leukotrienes, they are LT, A2, A4, B4, B1, B2, B3, and B4. I think it is there. D. All those leukotrienes are there as we have prostaglandins. What are their functions? They are also mediating in vasodilation and all. They are also mediating in immunity. One most important function for leukotriene is your bronchoconstriction. So in asthmatic people, leukotriene will be increased. So in order to decrease the asthma, we have to give leukotriene inhibitors. What leukotriene inhibitors we have to give? Like the xylutine and all. And also you can give leukotriene receptor blockers like montelukast. If anyone has asthma among you, okay, they will be taking what? They will be taking leukotriene inhibitors also. Montelukast, zafirlukast and all they will be taking. And one more important thing, some NSAIDs are contraindicated in your asthma. Why? Because if you inhibit the COX pathway, your entire phospholipid is now going towards LOX pathway, increase the leukotriene and they will cause asthma. Okay. Hope this is clear. All this you write, whatever I told in this prostaglandins, everything is important for biochemistry. Everything you write in the biochemistry, you will get good marks. Compulsory, you will get one question on prostaglandins. This is the leaked question. Biochemistry, prostaglandin is a leaked question. Compulsory, you will get one question. Immunology, compulsory, one question you will get. Innate immunity, you may describe all those barriers and all, anatomical, physiological, cells, proteins. Remember that story that I told? Okay, I tried to simplify how T helper zero cells and all, they are mediating the adaptive immunity, and what are the 5 processes by which the humoral and adaptive immunity is killing. Everything is now, I think, clear. I tried to simplify. This is taken in 15 to 16 sessions. I have not taken in much detail, but try to consolidate in one class. You will read much more detail in your pathology and your microbiology. For physiology, this much was needed. Hope there are no other doubts. If there are no other doubts, I think we can now leave. Okay. Please do fill the. अरे कोई तो भी feedback form डालो group पे. जल्दी बच्चे चले गए तो फिर करते नहीं.
डाल दी. Yeah, before all of you leave, I just wanted to say that maybe next week we'll have a genetic session by me. So genetic is a part of your anatomy paper, and it's very important. You'll definitely get, maybe 10 to 15 marks weightage will be there in your paper, because your paper is for 100 marks. Definitely 10 to 15 marks will definitely be from genetics, maybe more than that. So please be there for genetic session next week. It's very important, and it just completes one of the important parts of your anatomy, and you'll have time to study other topics if you just complete the genetics in the next week. So yeah, you can leave if ARBAZ has nothing else to say.
Yes, I don't have anything else to say. And regarding the further sessions, as Pranav told, next Wednesday, Wednesday, Thursday.
I'm not sure. Maybe in Monday or Wednesday.
Okay, 2 sessions will be taking for genetics. Be present over there. You will be getting all the notes for that. And next you'll be having eye physiology, ocular physiology, everything in one. And another session I think you'll be having ECG part 2. And one more session you'll be having regarding your auditory physiology. Renis Webers and all also will be explained in detail over there. Your practical also. And one more session I think you have regarding, many more sessions are there, but for this week and all, these are the sessions next week.
Yeah, tomorrow we don't have any session, right, ARBAZ? Someone asked if we have a class tomorrow.
No, we don't have any session tomorrow and day after tomorrow. Day after tomorrow I think Pranav may take, but tomorrow we don't.
Okay,
and one more very important session we have for acid-base disorders. Acid-base disorders also.
Would you like to tell about the YouTube one?
कुछ doubts है?
Or no? नहीं, डाउट नहीं है. वो जो तुम बनाए ना YouTube पे, तो बोलते क्या पूछ रही थी मैं?
क्या?
YouTube.
नहीं रखो, वो रखो, ये रखो.
तुम्हारी भैरी सूरत को सुन यार.
Okay, if you guys have no doubts, please leave the session. ARBAZ? ARBAZ?
I think he left.
Yeah, no, I'm there.
Yeah.
No, he's the organizer today. So please don't end the meeting. Let everyone leave the meeting. I have something to say.
Okay.
Okay, other than the panelists or everyone else, please leave the meeting.
Who were the panelists present today in the session? Only Yumi and Nusrat.
Yeah, I actually, wanted to see that who all are there, the panelists. I think सब लोग चले गए. Who is this? Safa, Shireen, and Harsh.
ARBAZ, I think you can excuse them. Okay, you have that option, right?
Yeah, you can excuse them otherwise.
Okay. I'm making you a Mexican organizer. I was taking the screenshots.
Pranav, you can end the recording.
नहीं मैं वो नहीं करा. ARBAZ बंद कर दो recording.
हाँ, sorry.